Welcome back. This is our second lecture, and in the previous lecture, DNA Fundamentals, we have discussed about purines and pyrimidines, the general concept, and I told you that in the second half, we will discuss about purine nucleotide synthesis and pyrimidine nucleotide synthesis. So in the first half, we will discuss about the purine nucleotide synthesis, and there are two types of synthesis. One is de novo synthesis, that is biosynthesis, and second one is purine salvage pathway. Let's discuss the de novo synthesis and move toward the board. You can see there is a ribose 5-phosphate. This is a ribose 5-phosphate. You can see this is the carbon number 1, carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and to the carbon number 5, there is a phosphate bond attached here. So from where does this compound actually comes from? Whenever you take carbohydrates from your dietary sources, they get metabolized in the liver, and this compound is present in a bulky amount in your liver. So what happens first? This compound reacts with ATP in a way that ATP gives us AMP, giving 2 phosphates, right? And this reaction is catalyzed by an enzyme that is PRPP synthetase. What happens? The 2 phosphates, they get attached to the carbon number 1 in a way that this is OHO and P of the first and O and second P, right? 2 phosphates, they came from ATP. So now this compound is known as phosphoribosyl pyrophosphate, PRPP, right? This is the rate determining step. This is the first step that we get PRPP from the ribose 5-phosphate. How? This compound reacts with ATP and make a word PRPP. So what happens? I told you in the previous lecture that in the purine is a double ring structure and having 4 nitrogen bases attached to it. So where does the nitrogens coming from? They came from the amino acids. So the first amino acid that gives its nitrogen is glutamine. So what happens? Glutamine gets converted into glutamate, giving us its amine portion, right? And this reaction is catalyzed by an enzyme that is PRPP glutamyl amidotransferase, right? So what happens? Now it will remove this POP portion and gets attachment of amine portion. Now you can see that, and here to the carbon number 5 there is a phosphate attached. Now you can see that this is phosphoribosyl and an amine portion. So this compound is phosphoribosyl amine, right? So this is the first step. Keep it in mind, that is very important, that first nitrogen is coming from glutamine. Glutamine is an amino acid, right? So first nitrogen coming from glutamine. What happens in the next step? Another synthetase enzyme comes, and it does what? When glutamine gives its first nitrogen, glycine gets jealous. Glycine says, if glutamine is giving its nitrogen, I'm going to get fully attached myself with this portion. So what happens? The whole glycine molecule get attached here. You know that amino acid has a formula that is alpha carbon. To one portion of alpha carbon there is amino group attached, to the other group there is carboxylic group attached. One side there is methyl group, and the other side there is hydrogen attached. In glycine, what happens? To the alpha carbon there is amino group, carboxylic group, and hydrogen, another hydrogen, the simplest amino acid. So what happens? The carboxylic portion gets its attachment to this portion in a way that you can see C double bond O here, CH2 and NH2. This is the amino portion. Now you can see that there is another nitrogen coming from glycine, and I told you that this reaction is catalyzed in the presence of an enzyme that is synthetase, and here the whole glycine is attached, right? Now you can memorize, keep it in mind that the second nitrogen is coming from glycine. The first one is coming from glutamine and the second one is coming from glycine. So what happens? In the jealousy, glycine proliferates itself. How it proliferates itself? There comes another reaction in the presence of an enzyme, that is formyl transferase. Formyl transferase. As the name indicates that formyl transferase will transfer formyl group. From which compound? That is very important. You have to keep it in a mind. N5 N10 methynyl tetrahydrofolate. When giving its formyl portion, it will convert into tetrahydrofolate, and its formyl portion will be attached to this portion, that is C double bond OH. This is the simplest formula of formyl group. Now you can see that here is the beginning of first ring, and I told you that in the purine there are 2 rings, right? So this is the beginning of first ring, and it proliferates itself. And now the glutamate thinks that glycine is proliferating itself in jealousy. I have to do something. It tells its parent, that is glutamine, that do something for me. Now what happens? The same reaction will repeat here itself. Another synthetase enzyme comes, and it will does what? It will attach another amino portion at this carboxylic acid oxygen place. So what will happen in the next step? The another amino group will get attached here, and this amino group is also coming from glutamine that is converted into glutamate, giving its amide portion, glutamate, right? And this amide portion is attached here. Now you can see there are 3 nitrogens, 1 nitrogen from glutamine, the second nitrogen coming from glycine, another amine portion from the glutamine. 3 nitrogen has been completed here. Now this nitrogen, this amine portion of the glutamine says to the glycine, let's join hand together. It's over. Let's over the fight. So what happens? To join hand, there is a formation of first ring, and keep it in a mind that whenever there is a ring formation, there will be a release of water molecule. So what happened here? That now you can move to this section. You can see that this is CH2, CH, H, and NH, H, NH2, and CH. Now this oxygen will be removed. I told you that whenever there is a ring formation, there is a removal of water molecule. So what will happen here? That this H and this H and the double bond O oxygen will make a water molecule. And now you can see that carbon has a valency to form a bond, and hydrogen of this nitrogen will move toward this portion, and this will become NH2. The single bond will form here. As you know that the nitrogen has valency of 3, so it will form 3 bonds, 1 nitrogen attachment with the carbon, second with the hydrogen, and second with the hydrogen. Now carbon will complete its valency by forming a bond here, right? And water will be removed out of it. This is a ring formation. Now you can see that there is 1 ring completed, and there is a beginning of second ring, right? So this compound is now known as imidazole. As there is a ring formation, now we will call it 5-amino-imidazole. I just wrote ribose 5-phosphate as it is intact throughout the process. Everything is happening above it, right? So this is now imidazole, and this is the initiation of a second ring here. Now you can see that there is 1 bond, second bond, and third bond with the hydrogen. So there is another valency, there is another space for a carbon to make a bond, right? So what happens the next process, next thing, that is carboxylation. Carboxylation reaction happens. What is carboxylation? As the name indicates, carboxylation means addition of carbon dioxide. So what will happen here? The carbon dioxide will get attached to this portion in a way that, as I told you, that this carbon is free and it can form a bond. Now the carbon dioxide will get attached to this portion, C double bond O and O, CO2, right? Now you can see that this is 5-amino-imidazole carboxylate, right? Carboxylation, after carboxylation, carbon dioxide is added to this imidazole, aminoimidazole, and this compound is now known as 5-aminoimidazole carboxylate. Right? So, this is very important. What will happen in the next section? There comes another enzyme that will attach to this oxygen another amino acid, that is aspartate. Right? Aspartate. Now you can check that the 3 nitrogens are already attached to this molecule. The first nitrogen coming from glutamine, the second nitrogen coming from glycine, the third nitrogen also coming from glutamine, and the fourth one nitrogen that is coming from aspartate. The whole aspartate will get attached here. I'm not going to write to confuse you people the whole formula of aspartate, but just amine portion gets attachment here, and to the alpha carbon at one end there is C double bond O negative, that is carboxylic group, another C bond O negative. I know that it's a little bit confusing, but the next enzyme will over your confusion in a way that the next enzyme is adenyl succinate lyase. This will end your confusion by removing the whole fumarate portion. Adenyl succinate lyase. It will remove the fumarate portion out of it, and only the amino portion will be attached to this portion. Now you can see that the 4 nitrogens in purine are completed. The first one from glutamine, the second one from glycine, the third one is also from glutamine, and the fourth one from aspartate. 4 nitrogens are now attached to this portion, and ribose 5-phosphate is intact throughout, right? Keep it in mind. Now you can see that the one ring has been completed, that is imidazole, and there is a beginning of second ring. Now what happens here, that to this portion, now the carbon's valency is satisfied here, to this portion one hydrogen will be removed and the nitrogen can form another bond. For what purposes? To make a second ring. So what happens at this position? At this position again the carboxylation will occur. Carboxylation will occur in the same way that tetrahydrofolate here, N10 formyl tetrahydrofolate will give its formyl portion. So what will happen here, that C double bond O H will attach here. This is again the formyl transfer, right? Formyl group is transferred here, right? How it is transferred? By N5 formyl transferase. So now you can see that carbon is ready to make a bond here to make a second ring. So in next half the ring formation will occur, and I told you in previous ring formation that whenever there will be a ring formation, there will be a removal of water molecule. So here there will come an enzyme that is cyclohydrolase. Hydrolase means removal of water. So this oxygen will be removed, and this hydrogen, and there is H2N. So one hydrogen from here and one hydrogen from here, they'll remove, and oxygen from here. So water will get out of it, right? So now the carbon will form a bond with the nitrogen, as the valency is not satisfied of nitrogen and of carbon too. So they will form a bond here by removing a water in the presence of an enzyme that is cyclohydrolase. And now you can see that the 2 bonds have been formed. 2 rings have been formed. The first ring, imidazole, and the second ring. Now you can see here, the first nitrogen, I am going to place this one to this place, coming from glutamine. The second nitrogen, that is this one, coming from glycine. The third nitrogen, this one, here this one, coming from also glutamine. And the fourth one, this one, is coming from aspartate. Now this structure, this basic structure throughout the whole process we get is known as inosine monophosphate. The story is not over yet. Now this inosine monophosphate is a precursor of, I told you that purine, pure as gold, purine, adenine and guanine. So it's a precursor of adenine monophosphate. Being a nucleotide, it will be a monophosphate. I told you that a nucleotide is a ribose sugar and a phosphate group attached to it and a nitrogenous base over it. So base has been formed. This is the precursor of this, that is ribose 5-phosphate. So this is a nucleotide. So now we can make what? We can make AMP, that is adenosine monophosphate, and the second one is guanosine monophosphate. How? Before going here, the necessary information for USMLE step 1, what you have to memorize from throughout the whole process, I'm going to write. No need to remember all these steps. This is just to give you a concept how these two rings structures form. Without giving this a whole concept, you cannot get to know what is the actual concept of it. So what will happen, what is necessary for memorizing it? There is this enzyme. This is a phosphoribosyl pyrophosphate, phosphoribosyl pyrophosphate glutamyl amidotransferase. So this enzyme is inhibited by a drug that is immunosuppressant drug azathioprine. Right? The azathioprine is an immunosuppressant, right? So azathioprine and 6-mercaptopurine. Basically, this azathioprine is a prodrug of 6-mercaptopurine. This enzyme actually inhibit this, these drugs inhibit this enzyme and will not let to proceed to the formation of purine synthesis, right? These drugs are basically given for the Crohn's disease. They are immunosuppressants to remove the inflammation of Crohn's. Due to the Crohn's disease, we give these drugs, right? So this is necessary information that you have to remember. The second one is the second thing that you have to memorize is here, nothing more. So the nitrogens, you have to remember that the first one nitrogen is coming from glutamine, the second one nitrogen is also coming from glutamine, the third one nitrogen coming from glycine, and the fourth one nitrogen coming from aspartate. This is the necessary information that you have to memorize. Now let's move toward how the adenosine monophosphate are derived from inosine monophosphate and how the guanosine monophosphate derives from inosine monophosphate. Let me remove this portion. So what happens here, if in the presence of GTP that will convert into GDP, giving us a phosphate bond, an enzyme that will react here is aspartate, another amino acid, aspartate. What will happen to this compound? You can see that there is a C double bond O. At this position, the O will be removed and the whole succinate portion of aspartate will get attached here. I'm not going to write everything here. Now I'm going to make a structure this way. Here's a nitrogen and here is another nitrogen and there is a double bond O. But now in the presence of GTP and GDP, O will be removed and here the whole portion of succinate will attach in a way that a amine portion NH to the other end is C alpha carbon and C double bond O negative, C double bond O negative. Now you can see that amine portion is attached here. This tension will be removed by another enzyme, and only amine portion will get attached here in a way that double bond, there's double bond, and this is also a double bond. Double bond and nitrogen. Now here attached is amine portion, and to this nitrogen ribose 5-phosphate. Now you can see that this is the basic building block, that is inosine monophosphate. What is the difference between inosine monophosphate and here is amine portion is attached to this position. If amine portion is attached to this position, this will become what? What is required? Pure as gold. So as portion has been completed, that is adenosine monophosphate. Adenosine monophosphate. Now you can see that how adenosine monophosphate has been derived from inosine monophosphate. What happens in the second structure, the second half, if there is a dehydration in a way that there comes an enzyme that is inosine monophosphate dehydrogenase, right? This will occur in the presence of NAD plus water, that will give NADH plus H2, right? So what will happen here? To this compound at this end, there will be nitrogen double bond, here, another nitrogen double bond, and this is another double bond. So what will happen at this end? Oxygen will be attached here. Now this is what, and this to this nitrogen, this is R5P. Now you can see that oxygen has been added to this place, and now this compound has become xanthosine monophosphate. Keep it in a mind, xanthosine monophosphate. From xanthosine monophosphate, we derive guanosine monophosphate in a way that another enzyme, that is very important enzyme, that is GMP synthetase, right? GMP synthetase. Now, GMP synthetase will do what? It will attach, it will convert glutamine. You can see glutamine is very important, giving us 2 nitrogen here, and in case of GMP guanosine monophosphate, giving another amine portion, that is now glutamine, giving us glutamate, and as a result we get what? Amine portion out of it. Now to this portion, in the presence of this enzyme, this amine portion gets its attachment here in a way that now you can see nitrogen, there is a double bond, another nitrogen, there is a double bond, and there is a double bond, and to this there is our R5P attached. And now at this point, here is an attachment of NH2. Now see that if inosine has amine portion on the top, it is adenosine monophosphate by this reaction, this whole reaction. And if another amine is attached here, this will become guanosine monophosphate. Guanosine monophosphate. Very important things now formed. That is, purine nucleotides has been synthesized. This is the de novo synthesis. right? This is the biosynthesis of this purine. Now, what to keep in mind here? The important things, the really important things. I told you that is necessary information for the purine salvage pathway. We are going to discuss right now after this. So you can see that if I remove this phosphate, it will become what? In the previous lecture I told you a phosphate group detached from nucleotide, what will happen? It will become nucleoside, right? If I remove this phosphate, or you can say that this phosphate is this one phosphate, right? If I remove this phosphate from here, it will not become adenosine monophosphate. It's no more adenosine monophosphate right now. It will become just adenosine, right? Adenosine, it's a nucleotide, right? So if I remove same phosphate from here, it is guanosine. If I remove phosphate from this inosine monophosphate, it will become what? Inosine. So what will happen if I remove the whole ribose sugar from here? The whole ribose sugar from here, the whole ribose sugar from here. Now they are just the free bases, right? Now this is adenine, not adenosine, adenine. This is guanine, and we don't call it inosine now because inosine was a nucleoside. This will now called as, after removing the whole ribophosphate, we will call it hypoxanthine, hypoxanthine, right? So I can say that if a ribose sugar is detached from guanosine monophosphate, the ribose is detached, this is a free base, this is known as guanine. If I remove this from adenosine monophosphate, this ribose sugar along with the phosphate, this is a free base known as adenine. And if I remove the precursor, the ribose sugar from the precursor, that is inosine monophosphate, if I remove the ribose sugar from it, it will called as hypoxanthine. I can get to know that if there are free bases present already, we just required what? I told you that it was the beginning of the step, that from first step, that is a amine group is getting attached from the glutamine, the whole step carried out. It is the end of whole step, right? So to make ribonucleotide, to make adenosine monophosphate, we just need one PRPP, right? We just need one PRPP. If one PRPP is added here, there comes an enzyme, we require one enzyme, that is ribosyl transferase, that will transfer this pyrophosphate region and attach to this portion, this whole portion, and will make it the required, you can say the nucleotide. If, for example, adenine is added here, this is adenosine monophosphate. If guanine is added here, this is guanosine monophosphate, right? So keep it in our mind for the purine salvage pathway. So let me erase this and start another one, that is purine salvage pathway. Take a break.